Okay, so energy production. For this, I'm just assuming that you have all probably had glycolysis and the Krebs cycle and the electron transport chain probably more times than you wish to have had to learn it. So we're just looking at the end result here. We're not going through every step of glycolysis and the Krebs cycle and the electron transport chain. It's just important to remember that glycolysis is the first step in carbohydrate breakdown. Glycolysis, like think glycogen, which is a long strand of glucose, lysis, which is breaking down, so the word literally is referring to carbohydrates. This would be happening in the cytoplasm, and it doesn't require oxygen. Not a really good source of energy. You're not going to get a lot of ATP production out of glycolysis by itself. What you are getting out of glycolysis is something called pyruvic acid, and that can be reacted with some coenzymes, we'll look at that in a second, so that it can go into aerobic respiration, which is the Krebs cycle and the electron transport chain that's happening in the mitochondria. So this requires oxygen. The Krebs cycle is a series of chemical reactions where we take what we've turned pyruvic acid into. So a product from pyruvic acid goes into the Krebs cycle, and we start rearranging carbons and things like that, and we end up giving off some ATP, and we end up with molecules that can go into the electron transport chain, and that's where we get the bulk of our ATP. We get lots and lots of ATP from the electron transport chain. So what we're going to look at here is how we get energy out of our three main nutrients: carbohydrates, proteins, or lipids. So for carbohydrates, we start out with glycolysis and we end up with pyruvic acid, and then we take the pyruvic acid and it reacts with coenzyme A. Coenzyme A, along with the pyruvic acid, is going to give us our product here, acetyl-CoA, and that's going to go into the Krebs cycle. So acetyl-CoA is what ends up going into the Krebs cycle, running through that and ending up with the electron transport chain and making plenty of energy there. Now for proteins, we have two kinds of proteins. There are glucogenic proteins and ketogenic proteins. Glucogenic proteins can be broken down into pyruvic acids, and then we just go right up to here. Same situation, it's just that we didn't use glycolysis because we're breaking down a protein or an amino acid, so it's a slightly different pathway, but we get still that final result of the pyruvic acid, which can react with coenzyme A, which can form acetyl-CoA, which goes into the Krebs cycle. So again, it's acetyl-CoA. Ketogenic proteins, on the other hand, are going to break into keto acids, which I'm sure everybody has heard of with all the keto diet stuff going around. But we all know that, ketosis is not a good thing because, being acidic is not a good thing. So your body has buffer systems to prevent you from going too much into ketosis. But this is a bad byproduct of breaking down a ketogenic protein, would be forming these keto acids. Now, in order to make the energy out of the keto acid, you react them with coenzyme A, and they turn into acetyl-CoA. So there's acetyl-CoA again. That's what's going to go into the Krebs cycle with ketogenic proteins or ketogenic amino acids. The downside for both of these, for the ketogenic ones, the keto acids, for both of them, there is ammonia formation. So that's toxic and needs to be detoxified by the liver. So that's not good. And also proteins are really valuable molecules. If you just think evolutionarily, proteins are the more difficult thing to get out of all three of these different nutrients. So it wouldn't be something you would want to waste burning for energy. It would be like if you were out in a cold woods or something and you had a bunch of paper and you also had a bunch of furniture. You're going to burn the paper, not the furniture, right? So the furniture would be the proteins in this case, and the carbohydrates would be the paper. You want to burn the paper, not the furniture. So you just don't want to use the proteins for toxic byproducts, ketoacidosis, and just that you need them for structural things and for creating enzymes and all that kind of stuff. So now let's look at lipids. We know we want to use carbohydrates over proteins for energy, but what about carbohydrates versus lipids? Now, lipids also break into keto acid. So if you do too much of that, there would be the danger of ketoacidosis, but you don't make ammonia as a byproduct, so that's good. You also get more energy per carbon. So if you had a lipid that was, say, 3 carbons long versus a carbohydrate that's 3 carbons long, you're going to get more ATP out of that lipid. So this is good for when you have time to make energy slowly. So you're just kind of sitting here, just watching a super fun A&P video. You're not really using a lot of energy, so you would be breaking down lipids preferentially because you have time to do it. Now, if you were to just start like running around or doing burpees or something, you need energy fast. Lipids are going to be too slow. You're not going to be able to get the energy fast enough to power through your burpees. So you need to use carbohydrates for energy in that case. So you use carbohydrates for quick energy, lipids for slow energy but more efficient. Okay, so we're going to finish up the digestive system with just talking about the liver really quick. The liver does a lot of different things. One, it does nutrient intro conversion, which we've touched on before. So I'm just going to remind you of what these things are. Glycogenesis, making glycogen. That's the stored form of glucose. Glycogenolysis, so glycogen, lysis, breaking down of glycogen. So taking glycogen and breaking it into its component parts, which would be glucose. Gluconeogenesis. Genesis means making, neo is new, glucose, so making new glucose. This would be making glucose out of a non-carbohydrate precursor, which is relatively easy for the body to do because glucose is really just carbons and hydrogen, so you can just pretty much make it out of anything. Deamination and transamination have to do with amino acids and proteins. So we know that proteins are just chains of amino acids in varying combinations. So if we are deaminating a protein, we're just taking amino acids off of it. That gives us keto acids as a byproduct, so not so good. That would be if we're using it for energy production, which we know we don't want to do. Transamination, we're just trading amino acids from one protein to another. So that is something, you have enough of this protein, but you don't have enough of that protein, so you can switch amino acids and create more of the protein that you need. So that is something that is a valuable thing to be able to do. Now your liver also makes bile, which we saw for buffering the chyme coming from the stomach, and we saw as helping us to absorb our micelles. Hepatocytes in the liver also make your plasma proteins like plasminogen, fibrinogen, the albumins, all these sorts of things that we have floating around, these proteins that are floating around in your blood made by the liver. And then they do detoxification, so various drugs, ammonia, metabolic byproducts, all sorts of stuff detoxified in the liver. Now, sometimes your liver can get damaged, and when your liver is damaged, that's called cirrhosis. This can be caused by hepatitis, which can be viral hepatitis, or it can be non-viral hepatitis. So you can have hepatitis from drinking too much alcohol. That will cause damage to your liver, which would cause cirrhosis. Or viral hepatitis, there's a whole lot of different ones of those. You've probably heard of hepatitis B and hepatitis C. Those would be the the kind that can be transmitted from person to person. C a little bit more so than B. Hepatitis A would be from like shellfish, eating, bad shellfish. So that would be something that you would ingest. And there's all sorts of different variations there of that. There's also cirrhosis that can come from a condition called hemochromatosis. And hemochromatosis is where you have cells in your liver called Kupffer cells. And these Kupffer cells are phagocytes. They also store iron. So in hemochromatosis, this is a genetic disorder where your Kupffer cells store too much iron, and that iron then damages them, and it escapes from them after it damages them, and it damages your hepatocytes, so you can't do all of this. And then it can get out into the blood and damage all the rest of your organs as well. So this is something that adds up over time as you get older. But it's a pretty rare genetic disorder, although my father has it. I might have it too. I don't know. I haven't been tested yet, but it's possible. But it really just means that you have to watch your iron intake. You have to donate blood if you have this to help you get rid of some of that excess iron. Anyway, the symptoms for cirrhosis are very similar, whether it's for viral hepatitis or shellfish-related hepatitis or alcohol-related hepatitis or from hemochromatosis. One, jaundice, where you look yellow, especially in, say, the whites of your eyes. And this is from a backup of bilirubin, because your hepatocytes are damaged. They can't use the bilirubin to make bile. They're not functioning properly. Next comes ascites. Ascites is where you get water building up between your visceral peritoneum and your parietal peritoneum. So between the body wall and the organs. So you get fluid getting trapped there or pushed in there, and that's because you have a lot of pressure in your hepatic portal vein when you have cirrhosis. It's like a backup of blood in the hepatic portal vein. And that just kind of pushes the fluid back into the abdominopelvic cavity and pushes it out of the cavity and into that peritoneal space. And then we have edema, which is the last symptom. Edema is where you get swelling, particularly in the periphery, so your ankles and feet, wrists and hands. And this happens because your B cup goes down. And if you remember your blood clot osmotic pressure, that was something that held fluid inside a capillary when we were talking about filtration pressures and all that. It lowered your filtration pressure. So those that blood clot osmotic pressure is due to plasma proteins. So if you're not making plasma proteins, this is going to go down. So you don't have that kind of check on filtration pressures, and you would just tend to have more filtration. So you get more water moving out of the blood and into the tissues. So these would be the symptoms for all of the types of cirrhosis. But if you had hemochromatosis, you would also have high levels of iron in your plasma. So that would be the kicker. That would be how you would know that it was hemochromatosis as opposed to another sort of cirrhosis. Okay, so that is it for the digestive system. That's it. Enjoy. See you later.